Okay, so we're going to be learning about DNA replication today. DNA replication is what occurs in interphase during the S phase. So we've talked about what happens in interphase, and you have G1, S, and G2. During the S phase, S stands for synthesis, you have DNA replication. So we are going to start off with kind of what we've been learning the last couple days, or it is what we've been learning, the structure of DNA. So this is like one last chance, or I mean, we'll learn again, but it's another chance to learn it. Nucleic acids are built from small monomers called nucleotides. Yesterday I had multiple students, number one, ask me like what was the monomer of a nucleic acid. If you're still having difficulty with that, that is part of two different standards. So nucleic acids are made up of nucleotides. When we did that DNA activity, each one of those things that you were cutting out was a nucleotide. Your nucleotide then consists of three parts: a sugar, specifically that sugar, which is right here, is called deoxyribose. It's literally in the name. Phosphate group, and a nitrogen base. The nitrogen bases, we have four: adenine, thymine, guanine, and cytosine, and these are their abbreviations. Take a second and try to say these out loud to yourself. That will help you to remember. Also, the phosphate group in the sugar will be the sides of the ladder, and the nitrogen base will be the rung of the ladder, and that was a question yesterday, and you guys literally built this. Okay, so there's base pairing rules. Adenine will always pair with thymine, so AT, and this is vice versa, by the way, guys, so thymine will also pair with adenine. Guanine is always paired with cytosine, so you have your A and your T together, and you have your G and your C together, and they will bond with a hydrogen bond in the middle. So you have these two, adenine and thymine, bonded with a hydrogen bond, guanine and cytosine bonded with a hydrogen bond. I always remember like the word at, and then G and C both have that same like curve in the shape of the letter, so they look the same. Another student had something, and he'll I'm talking about, but the apples in the tree, and then the grapes in the cytoplasm, and also another student said the grapes in the cellar. I don't know. There's a lot of different things that you guys can think of to help you remember. Okay, so now we're to the new stuff. So DNA replication. Before a cell divides, and just so you guys know, every time you see cell division, that's mitosis. It duplicates its DNA during the S phase. So this is where you go from a chromatid to a chromosome, and that occurs in the S phase right before you go into into cell division. Okay, so that's when you go from 1 to 2. So that's what we're talking about here. We're in the S phase. So while it's in the S phase, step 1, so yesterday you guys built this. So imagine those staples breaking. So you use the staple to create the hydrogen bond that bonds the two bases. So the adenine and thymine, and the guanine and cytosine. So the two strands of each DNA molecules will separate by an enzyme called helicase. So helicase is an enzyme. It's going to come, remember, enzymes help catalyze and speed up reactions. This is the reaction that's occurring. The enzyme will unzip. Think about a zipper, or just imagine like all those, staples coming off, breaking the hydrogen bond and creating, it's called a replication fork. So right here, this is the fork, right here. So the separation is the fork. So step 1, you unzip. Imagine again all those staples coming apart. Step 2, we're going to make a new complementary DNA strand. The reason why they call it a complementary strand is because you're going to be, and I'll get to this in a minute. So by the way, this is your replication fork. See it right there? And this is the helicase. So the two new complementary strands are synthesized following the rules of base pairing. So this is what I was trying to say earlier. So this has been separated by helicase already. And then what happens is you have a new, so once the separation occurs, you're going to recreate it with the pair. So like if there was an A there, you'll make a T. And we'll practice this, guys, so don't worry. 2 new complementary strands are synthesized following the rule of base pairing, adenine pairing with thymine and cytosine pairing with guanine. Each strand of the double helix of DNA serves as a template for the new strand. So again, if you're reading a thymine, then it'll make a cytosine. A thymine, it will create an adenine. If it's a cytosine, they'll create a guanine. They create the complementary one. And again, we're going to actually recreate this using a foldable. So DNA polymerase, this is another enzyme. So there's 2 enzymes we just talked about, helicase, and the other one's called DNA polymerase. This enzyme called DNA polymerase will attach to the new strand and join the nucleotides to synthesize a new complementary strand of DNA. So when it breaks apart, this DNA polymerase will attach and read, okay, adenine, we need a thymine, boom. Guanine, we need a cytosine, and it'll just like read it and then create the complementary strand so that you have like that new DNA. So you have one being made down here and one being made down here essentially. So that one, and there's your fork, and then you'll have that going on. The enzyme also will proofread each new DNA strand to make sure it's like a perfect copy. That's it for the guided notes portion, but we're not done yet. We're actually going to watch a quick Okay, so we're going to be done now. It did go a little bit more into depth with things you don't need to know at the end, but if you were wondering what happened to the other strand, it kind of goes the opposite way in a lagging formation. So, but that's it. All right, so great job.